So these vaccines and viruses that they're making up in the labs and they're sitting on their shelves, and the new ones can be placed in food or basic medicines, and they can be deployed that way. So we wouldn't even know it. You could take your prescription and be infected. Food, and then you have the Monsanto. They knew how terrible Monsanto was with their using viruses to create food. I mean, that's how the Monsanto seed is. It's a virus. And they put all kinds of crap in there. I mean, I heard that somebody won a case or something, but is Monsanto been kicked out of the U.S. for the monster foods? I mean, they could be putting these pandemic flus, viruses in the Monsanto monster foods, and it can develop into a pandemic, infect animals. Could be what happened to those deer. They call it zombie deer, and once again, scientists are baffled. They're baffled. Gosh, it's just unbelievable how much they lie to us, how much they want to kill us.
52. Influenza biological. Pandemic influenza vaccine phase 3. Medline plus related topics. Bird flu. Drug information available for flavirin influenza vaccines. U.S. FDA resources. Study type. Interventional. Study design. Prevention. Randomized. Single blind. Active control. Parallel assignment. Safety study. Official title. A phase 3. Randomized. Controlled, observer blind, multi-center study to evaluate the immunogenicity, safety and tolerability of 2 doses of FLUAD H5N1 influenza vaccine in adult and elderly subjects. Further study details as provided by Novartis. Primary outcome measures, adverse event rate. http://clinicaltrials.gov/ct2/show/ncT004347333. Secondary outcome measures. Seroconversion and seroprotection after 2 doses of H5N1 vaccine. Estimated enrollment, 4,400. Study start date, January 2007. Eligibility, ages eligible for study, 18 years and older. Genders eligible for study, both. Accepts healthy volunteers, yes. Criteria, inclusion criteria, healthy subjects 18 years of age who signed the informed consent. Exclusion criteria, receipt of another investigational agent within 4 weeks. Receipt of influenza vaccination for current season 2006-2007. Any acute disease or infection, history of neurological symptoms or signs, known or suspected impairment of immune function, any serious disease, bleeding diathesis, fever, defined as oscillatory temperature cubed 38.0 degrees Celsius, within 3 days, prior to visit 1.
assassinateevilnow@gmail.com if anybody wants a copy of this. And they will do their best, not me, because I'm just the narrator, to get the reports to anyone who requests them. That's been on the previous videos too, so be patient with them. They will get around to it.
53. pregnant or breastfeeding or females of childbearing potential who refuse to use an acceptable method of birth control. Surgery planned during the study period. Hypersensitivity to eggs, chicken protein, chicken feathers, influenza viral protein, neomycin or polymyxin or any other component of the study vaccine. Receipt of another vaccine within 3 weeks prior to visit 1 or planned vaccination within 3 weeks following the last study vaccination. History of or current drug or alcohol abuse. Any condition which, in the opinion of the investigator, might interfere with the evaluation of the study objectives. Contacts and locations. Please refer to this study by its clinicaltrials.gov identifier, NCT00434733. Locations: Poland. Centrum Badań Farmakologii I Clinics na Monopol, Krakow, Poland, 30 to 969. Sponsors and collaborators: Novartis, Novartis Vaccines. Investigators: Study Chair, Novartis Vaccines and Diagnostics GmbH & Co. KG. Novartis, Novartis Vaccines and Diagnostics GmbH & Co. KG, Germany. More information: no publications provided. Study ID numbers: V87P4 2006 00542818. Study first received February 12, 2007. Last updated April 23, 2008. clinicaltrials.gov identifier, NCT00434733. History of changes. Health Authority, Poland, Central Register of Clinical Trials, CEBK. Keywords provided by Novartis. Influenza H5N1. Pandemic. Study placed in the following topic categories: virus diseases, influenza, human, respiratory tract diseases, influenza in birds, respiratory tract infections, orthomyxoviridae infections. Additional relevant mesh terms: virus diseases, respiratory tract infections, RNA virus infections, influenza, human, respiratory tract diseases, orthomyxoviridae infections. clinicaltrials.gov processed this record on May 17, 2009. '54. Back to top of main content. U.S. National Library of Medicine. Contact Help Desk, U.S. National Institutes of Health, U.S. Department of Health and Human Services. When damage and injury, however, are listed as the primary outcome, this is no longer medicine. This is murder. Any vaccine for a pandemic influenza should have to be thoroughly evaluated through trials and research to prove its safety, efficiency, quality and beneficial health effects. If a government is going to be in compliance with its duty under normative justice to issue a license for that vaccine. Moreover, vaccines and drugs should have been tested for their beneficial health effects in several clinical phases for safety and efficacy before they can be released to the general public. This is a time-consuming process, often taking years to complete. There is no shortcut to following these procedures when it comes to safety. Any new vaccine has to be evaluated at many levels: Phase 1, safety; Phase 2, safety and immunogenicity; Phase 3, large-scale trials for efficacy; and Phase 4, post-marketing surveillance. It is criminal for a vaccine material that has as its stated primary desirable outcome, adverse events rate, after 2 doses rather than positive events rate, that is, beneficial effects on the health of the patient, to be injected into patients. It is a crime to produce a vaccine whose overwhelming intention is to produce adverse events or damage to the people who are injected with the drug, as the FLUADH5N1 does. It is a crime to approve that vaccine for the market on the basis of it producing adverse events rate. If I make a drug saying its success is measured in terms of adverse events, and to damage people, I am conspiring to commit premeditated assault or murder using a bioweapon and an injection as the delivery system. If I actually use that drug and kill people, I have committed premeditated murder using a bioweapon and an injection as a delivery system. The doctors and nurses involved in the bird flu trials in Poland are now on trial for having withheld from their victims information about the drug, presenting it instead as a harmless, routine shot. Insofar as they have violated the requirement to obtain informed consent, they have violated the medical law. Insofar as their actions led to the deaths of others, they have violated criminal law. Are the people of the United States going to be forced to take an unproven, untested vaccine such as the one produced by Novartis, fully licensed but licensed to cause adverse events, that is to say, to kill and injure? Novartis, along with Baxter, is one of the two major companies with contracts to produce millions of doses of swine flu vaccines for a mass compulsory vaccination. Novartis has also received contract from USDHHS to further develop MF59C1 adjuvant technology to potentially extend vaccine supplies in case of influenza pandemic outbreak. 55. http://www.antiviralintellustrate.com/1/database. Prod equals 1737. Compana market cap 2009p, E5-year earnings growth technology, Novartis, NVS, $85 billion 10 by 10% cell-based vaccines, Baxter, Bax, $30 billion 13 by 12% cell-based vaccines, Gilead, Guild, $40 billion 18 by 15% antiviral drugs. Crucell, CRXL, $1.5 billion 50 by 30% cell-based vaccine. Gilead will receive royalties on every dose of Tamiflu sold by Swiss-based Roche. The current efforts to beef up emergency stockpiles of Tamiflu could add $80 million to Gilead's bottom line within 2 years. Novartis, Baxter, and Crucell are each developing vaccine production methods to replace our antiquated system, which uses chicken eggs. From start to finish, each of the new approaches can generate an original vaccine within 12 to 16 weeks. Novartis already has the genetic code of the current swine flu virus. Now, it's waiting for an actual sample of the virus to arrive in its labs. Baxter expects a sample, as well, in the next few days. Evidence as to the WHO's role in the bioweapons program. The World Health Organization, WHO, is a specialized agency of the United Nations, UN, that acts as a coordinating authority on international public health. Established on the 7th of April 1948, and headquartered in Geneva, Switzerland, the agency coordinating international efforts to monitor outbreaks of infectious diseases, such as SARS, malaria, and AIDS. WHO is currently working with Collaborating Center in Atlanta, the Centers for Disease Control and Prevention, CDC, in the United States of America, and vaccine companies such as Baxter and Novartis to develop candidate vaccine viruses for 4 billion people by autumn of the world's population, enough to achieve an 80% reduction in the world's population. There is evidence that WHO itself is playing a role in exposing the populations of the world to the risk of a pandemic virus that could kill billions of people. Though Dr. Margaret Chan, the Director General of WHO, is technically a public servant and has the duty as part of her official capacity to act at all times in such a way as to safeguard the health of the world's population, there are grounds for believing WHO is abusing its administrative structures, personnel and services, actually misusing pandemic material and pandemic declarations to assist organizations, companies, government bodies or other entities intent on unleashing a pandemic virus and then carrying through a mass vaccination program with contaminated material in order to gain political and economic advantages from mass murder. WHO supplied the wild bird flu virus from its reference laboratory that Baxter AG 56. Austria then used to produce 72 kilograms of contaminated bioweapon material that nearly